Hello, my name is Robert, and I am here with Molly, who has offered to help me learn about lysosomal storage diseases, or LSDs. So, I understand there are many LSDs that can cause a wide variety of impacts, and these conditions are genetic, so people are born with them. Which LSD do you have?
I inherited Fabry disease from my mother.
I know understanding genetics and cell biology can be difficult, so I thought it might be easiest to go straight to the source, your cells.
That's a great way to get the whole story.
What causes lysosomal storage disease? Our bodies have organs like lungs, heart, kidneys, intestines, and more to perform specialized functions. Likewise, cells have organelles to perform specialized tasks on the cellular level. I've heard the nucleus is the brains of the operation, so let's start there. Hello, nucleus. Can you tell me what causes a lysosomal storage disease, or LSD?
Well, I guess the simplest answer is a LSD is caused by a change in a section of DNA. I contain the chromosomes, which are made up of long chains of DNA. This DNA is the body's blueprint or instructions for how everything is built and functions. Sections of DNA are divided up into genes. Genes control lots of things. You get half of your genes from your mother and half from your father. This system works great most of the time, but sometimes disease-causing changes happen in genes. With different LSDs, different genes are involved. Sometimes a change in just one gene causes the disease, but others may result from changes in multiple genes. If I were you, I'd talk to the endoplasmic reticulum next. He goes by ER, and he can tell you more.
Thanks for all the great information. Hello, ER. Tell me about yourself.
Well, I use gene instructions to create proteins. I follow the genetic instructions exactly.
Okay, so what can you tell me about the genetic errors that cause lysosomal storage diseases?
When genetic differences happen in the instructions, different diseases can result, including LSDs. The proteins or enzymes involved for LSDs are used to break down certain substances into smaller pieces or for protein transport. I do quality control checks on the protein chains I make, and when they are changed too much, I can get rid of most of the broken enzymes. Simply switching two pieces of code may seem like a little change, but the impact can vary from small to big. All these types of genetic differences can result in non-functioning or misfolded, unstable enzymes. You should talk to the Golgi body. GB knows where enzymes go next.
Thanks for explaining what you do. Hello, Golgi body. I'm doing a story about lysosomal storage diseases. What role do you play regarding enzymes in the lysosome? My main job is to check what I receive from the endoplasmic reticulum and deliver it to the correct place or organelle, such as the lysosome. When an enzyme contains a difference that changes its shape, it may not get to me, and of those that do, they often don't work right, even if I am able to send it to the lysosome. Thanks. That helps me better understand your job better. It's finally time to talk to the lysosome. Hello, lysosome. I am working on a story about lysosomal storage diseases, and I'm investigating the various steps that can cause an LSD. What can you tell me about these diseases? LSDs are a group of over 50 rare conditions that occur when a specific enzyme is missing or doesn't work properly. The enzyme's job is to break apart or degrade a specific substance in the cell that is no longer needed. I am the special place where the conditions are right for enzymes to do their work. For example, there is a substance or substrate known as GB3 made of a fatty acid unit. Attached to 3 sugars, and the enzyme alpha-gal A breaks off the last sugar unit. Other enzymes can then break off the remaining sugars, and in this way, the substrate is degraded into small enough pieces so the cell can then reuse them or get rid of what is not needed.
If the necessary enzyme is missing or unable to do its job, the substrate builds up like trash bags you can't remove from the kitchen. And it can cause the cell to not function correctly or even die. The buildup of GB3 does this in Fabry disease. Some of the LSDs affect most cell types throughout the body, while others may affect only certain cells. The speed at which substrate builds up may vary as well. There can be a wide range of symptoms from one disease to another, or even within the same disease.
It sounds like LSDs can be very complicated.
They certainly can.
How are LSDs inherited?
I'm a certified genetic counselor, and I can answer that. All lysosomal storage diseases are inherited from one parent or both. They are either autosomal recessive or X-linked. Only a few are X-linked. Autosomal recessive diseases like Gaucher or Pompe are passed on to children when both parents are carriers and contribute their affected chromosome. For each pregnancy, there is a 25% chance for this to happen. There is a 50% chance for the child to inherit the gene from only one parent and be a carrier. There is a 25% chance the child will not inherit the gene at all. Passing on a particular gene is not a choice. It is no different than the trait for black hair or green eyes. X-linked conditions like Fabry or MPS type 2, also known as Hunter disease, are passed on by the X chromosomes. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. Any children of a female with an affected X have a 50% chance of inheriting the affected chromosome. This is the case no matter the sex of the child. A male with an X-linked condition will pass his affected gene to 100% of his daughters, since the genetic difference is on the X chromosome. His sons have a 0% chance and will never be affected since they receive his Y chromosome.
That really helps me so much, and I'm sure it helps others too. Thank you for your time.
You're welcome. Thank you for having me. To learn more about genetics, testing, risks for family members, and possible treatment, please contact a geneticist or a certified genetic counselor.